Ephedra introduction. Ephedra is an evergreen shrub-like plant native to Central Asia and Mongolia. It is considered as advanced type Amygymnosperms because of the presence of vessels in xylem tissues and inflorescence like male and female strobili. Vegetative features of Ephedra. The vegetative plant body contains roots, stem and leaves and shows xerophytic characters. Shrubby plant body usually remains less than 2 meters in height in most of the species. It resembles a quasitym in its external morphology. A prominent underground tap root system with many adventitious roots is present in Ephedra. Roots contain many root hairs but there is no mycorrhiza. The stem is green, ribbed, profusely branched, hard and glabrous, and bears nodes and intermodes. It is distinctly jointed. The stem, being green, performs the function of photosynthesis. The leaves are small, scaly and rudimentary. They are present in opposite decussate manner, or in whorls of 3 or rarely 4. There is hardly anything which could be called a blade in Ephedra. Scaly leaves fuse at the base to form a basal sheath. Each leaf contains 2 unbranched, parallel veins. A bud, which forms the branch, is present in the axil of each leaf. True foliage leaves are absent in Ephedra. Anatomy of Ephedra. Young stem. The outline shows many ridges and grooves. Epidermis is thickly cuticularized. The epidermal cells are thick-walled. Patches of sclerenchyma are present, particularly below the ridges. Continuity of the epidermis is broken by many sunken stomata present in the grooves or along the sides of the grooves. The wide zone of thin-walled, chlorophyll containing green cells is present in between the thick-walled sclerenchyma and the vascular cylinder. Many intercellular spaces are also present in this region. In some species this green region is differentiated into palisade and spongy parenchyma. Some sclerenchyma patches are also irregularly distributed in this green region. Endodermis is clearly demarcated only in very young stems. Pericycle is not clearly defined. The vascular cylinder is an endoxiphonostele. It is formed by an interrupted ring of 8 to 12 conjoint, collateral, open and endarc vascular bundles. The number of the primary vascular strands is generally 8. Out of which 4 represent the foliar traces while the remaining 4 are stem bundles. Foliar traces run up to the node. Each leaf receives 2 bundles. The nodes are in a lacunar and 2 trace type. In each vascular bundle the xylem is present towards inner side while the phloem towards outer side. The xylem consists of trichates, vessels and xylem parenchyma. The vessels contain bordered pitted thickenings. The trichates may be annular or spiral. The phloem consists of sieve cells, phloem parenchyma and albuminous cells. The albuminous cells look like the companion cells of angiosperms. A well-developed parenchymatous pith is centrally located. It becomes thick and sclerenchymatous in the older stems. Tannin-like contents are present in a group of thick-walled cells in the center of the pith. Old stem showing secondary growth. Vascular cambium, responsible for secondary growth, is present in between primary xylem and primary phloem. After forming a complete ring of cambium, the latter cuts secondary phloem towards outer side and secondary xylem towards inner side. The distinct annual rings are formed because of the varied activity of cambium in different seasons. The secondary xylem consists of vessels, trichates and small amount of xylem parenchyma. Fibers are absent. The secondary phloem consists of sieve tubes and companion cells as in netum. These are formed in rows. Some albuminous cells are also present in the secondary phloem. The presence of vessels is the most striking anatomical feature of Ephedra. Vessels are modified trichates of all sizes. The largest vessels are formed in spring but in the later part of the season the size decreases. The bordered pits are present. After some time they become simple perforations by losing the torus and border of the pits. In TLS medullary rays are clearly demarcated. They are very long, wide and uniseriate, but become multi-cennate by the longitudinal divisions. The rays consist of isodiamithic, elongated or curved cells, the size of which varies. Their walls are thick, linified and pitted. The pits are simple and small. In Ephedra the wood, in general, is hard because most of its cells are thick-walled. In RLS large vessels and many trichates are seen. The trichates show bordered pits which are usually uniseriate and more on the radial walls. Vessels are the most prominent features. The pith becomes narrow in the stems undergoing secondary growth. It becomes very scanty in old stems. Leaf. In TS the reduced and membranous scaly leaves are somewhat oval in outline. The epidermis consists of elongated or oval cells. Cuticle is also present. Stomata, when present, are sunken. 2 to 3 or more layers of chlorophyll containing cells of palisade tissue are present. Major remaining part of the scaly leaf remains filled with cells of spongy parenchyma. Many air spaces are present in the parenchymatous and palisade regions. The vascular bundles are 2 in number. They are small and remain embedded in the parenchymatous tissue. Reproduction of Ephedra. Ephedra is dioecious, and the two sex organs are present on different plants. Ephedra is heterosporous, i.e., two types of spores, microspores in male flowers and megaspores in the female flowers, are present. The male and female flowers are present in the form of cone-like, compound male and female strobili, respectively. It is only at this stage that the distinction between the male and female plants can be made because there is no well-marked morphological difference in the vegetative stage of the male and female plants of Ephedra. Male strobilus. The male strobili are compound structures arising in clusters from the nodes of the branches. Each strobilus develops in the axil of a scaly leaf bract. A male strobilus is a round or ovoid body with a strobilus axis in its center. 2 to 8 pairs of bracts remain arranged in opposite decussate manner on the strobilus axis. All the bracts are fertile except a few on the lower side. A single male or staminate flower arises in the axil of each bract. Each male flower consists of 2 bracteoles and a stamen. Bracteoles are thin opposite scales united at the base. They have been interpreted as periant. Each stamen is a stalk structure. The stalk continues into a short axis or microsporangiophore which bears 2 to 8 or more microsporangia terminally. Microsporangia are sessile structures. Each microsporangium is bilicular or trilocular. The male flower is called a simple strobilus. Development of microsporangium. The microsporangium starts to develop from a group of hypodermal cells which function as archosporium. The archosporial cells are larger in size with more dense cytoplasm and quite prominent nuclei in comparison with the other adjacent cells. These cells divide pericolinally into outer primary parietal cells and inner primary sporogenous cells. The parietal cells divide repeatedly and form one cell thick wall and also the tapetum. The sporogenous cells divide several times irregularly and form many microspore mother cells. The latter divide meiotically to form spore tetrads arranged tetrahedrally. The haploid microspores are later on separated. Rarely, the spores are arranged isobilaterally. Female strobilus. The ovulate or female strobili are elongated and pointed structures. Similar to male strobili, they also develop in the axil of leaves in the whorls of 2, 3 or 4 at the nodes of small green branches. Each female strobilus is sessile and smaller than male strobilus. Pairs of bracts are more in number in female strobilus than male strobilus. Bracts are arranged in opposite decussate manner. All bracts, except the uppermost pair, are sterile. 2 ovules are present in the axil of uppermost pair of bracts, out of which generally only one survives. The female strobili appear on the plants in the month of April and the mature seeds are seen in September in Ephedra gerardiana. Ovule. The ovule remains covered by a cup-shaped outer integument and an inner integument. The outer integument remains attached at the basal portion of the ovule. The inner integument protrudes out in the form of a long tubular micropyle. The integuments enclose the nucellus. A small pollen chamber develops near the micropyle in the nucellus. Pollen chamber in Ephedra is deepest known among the gymnosperms. The female gametophyte is centrally located and the Archegonia are present in the female gametophyte near the micropylar end. The hostorial region is present near the chalazal end and bears some hostorial processes. A hypodermal archosporial cell gets differentiated in the parenchymatous nucellus of the young ovule. This archosporial cell divides pericolinally into outer parietal cell and an inner megaspore mother cell. The megaspore mother cell is pushed quite deep into the nuclear tissue and divides meiotically to form 4 haploid megaspores. The archosporial cell functions directly as a megaspore mother cell in Ephedra intermedia. Microsporogenesis and male gametophyte. The microspore is the first cell of the male gametophyte. It is wingless, unapicturate and has a thick axene. The germination of microspore starts within the microsporangium. At the time of germination, the microspore elongates and divides to form a prothelial cell. The second division results in the formation of a second prothelial cell. In Ephedra trifurca, the second prothelial cell is not separated from the antherodial initial by a wall. The generative cell soon divides into the nuclei of stalk cell and body cell. A common mass of cytoplasm surrounds the nuclei of stalk cell and body cell, and they are never separated by a cell wall. Pollens are shed at this 5-cell stage and are carried up to the micropyle of ovule with the help of wind. Upon reaching the ovule, the axene of the pollen grains ruptures and the intine comes out in the form of a tube. The generative cell divides and forms 2 male nuclei. Female gametophyte. The functional megaspore is the first cell of the female gametophyte. It enlarges and its nucleus divides several times by many free nuclear divisions to form as many as 256 nuclei in Ephedra trifurca and 500 nuclei in E. foliata. The free nuclear divisions continue for about 20 days. The nuclei get themselves arranged around the central vacuole on the periphery of the megaspore. The free nuclei are evenly distributed throughout. The wall formation starts from outside, proceeds rapidly towards the center, making the complete structure ultimately cellular. In this cellular female gametophyte, the cells of the upper reproductive region are large and elongated as compared to that of the lower nutritive region. The nutritive region gets further differentiated into upper storage region and lower hostorium. Archegonia develop in the micropyle region. Archegonium. The number of Archegonia in Ephedra varies from 1 to 3, but their usual number is 2. An Archegonial initial divides pericollinally to form an upper primary neck cell and an inner central cell. The primary neck cell divides several times to form 4 to 5 or more tiers and then appear anticollinal divisions. More than 32 neck cells are formed after anticollinal divisions. Due to certain irregular divisions in neck cells, its tissue sometimes becomes undistinguishable from the surrounding cells of the female gametophyte. The archegonial neck in Ephedra may sometimes become as long as 40 cells. The central cell enlarges and its nucleus divides into a ventral canal nucleus and an egg nucleus. There is no wall formation between these two nuclei. The ventral canal nucleus may or may not move down towards the egg nucleus. The cells adjacent to central cell may divide transversely to form a clear 2 to 3 layered jacket. A mature archegonium thus consists of a long multilayered neck and a central cell containing a ventral canal nucleus and an egg nucleus. The long columnar neck appears similar to the transmitting tissue in the style of angiosperms. The archegonium in Ephedra is rich in protein but poor in polysaccharides and RNA. Fertilization. At the time of fertilization, the pollen tube penetrates the archegonium and discharges its contents into the egg cytoplasm. One of the 2 male nuclei fuses with the egg nucleus and forms an oospore or zygote. The other male nucleus may fuse with the ventral canal nucleus, exhibiting the phenomenon of double fertilization. The second fusion, however, does not result into any embryo formation. Chamberlain, 1935, however, stated that whether there is any double fertilization, like that in angiosperms, is questionable. Land, 1907, however, opined that there is no definite fusion of the second male nucleus with the ventral canal nucleus in Ephedra. But Hersfeld, 1922, observed that there is a clear-cut double fertilization in E. campylopodà as in angiosperms. Embryogeny. The zygotic nucleus undergoes free nuclear divisions, resulting into 8 free nuclei which remain evenly distributed throughout the cytoplasm. A cell gets organized around each of these nuclei, and each of such cell functions as a potential pro-embryo. A tubular outgrowth, called suspensor tube, develops from the pro-embryo. Its nucleus also divides simultaneously into 2 nuclei, i.e., embryo nucleus and suspensor nucleus. The wall between the 2 nuclei is formed. The embryo nucleus passes into the tube, which develops continuously and carries the lower embryonal cell deep into the female prothallus. The embryonal cell divides and develops into the embryo proper, which contains 2 cotyledons. Seed. The seed contains a dicotyledonous embryo. It remains situated at the tip of the elongated suspensor and remains embedded within the tissue of the female gametophyte. The remnants of nucellus are seen in the form of a disorganized sheath of cells. The seed coat consists of two separate layers derived from two layers of the envelope. At maturity, the subtending adjacent bracts of the strobilus in Ephedra foliata become thick and fleshy and form an additional covering over the seed. Thus, the seed remains covered by three envelopes. Seed germination. The seed germinates without any resting period and the germination is of epigeal type. Sometimes the seed may even germinate within the parent strobilus. The cotyledons grow steadily until they become several centimeters long.